Cancer, a disease of aging, is a group of diseases characterized by uncontrolled growth of abnormal cells. You might picture cancer as a lump, a mass, a mutated cell, or even something aggressively spreading throughout the body. But today, we'll dive deeper into the biological world of cancer. As normal cells transform into cancer cells, they gain at least one of the hallmarks of cancer: sustained growth, evading tumor suppressors, resisting cell death, immortality, angiogenesis, metastasis, and later expanded to include genomic instability, altered metabolism, evading immune system, and tumor-promoting inflammation. Our focus today is on one hallmark that is responsible for about 90% of cancer-related death: metastasis. What is metastasis? Metastasis is the spread of cancer cells from the place where they first formed to another part of the body. The journey isn't random. It follows a defined sequence called invasion metastasis cascade. We can break that process into 6 main steps: local invasion, intravasation, transport, trapping of circulating tumor cells, extravasation, and lastly, colonization. I know it sounds complicated, so I'll give an analogy before getting into the details. Imagine you're driving home to spend the Christmas holiday with your family. You start by preparing for the trip, packing, putting on your coat, and heading to your car. This mirrors tumor cells breaking through the surrounding tissues to escape the primary site. Once you reach the car and you start driving, you eventually get onto the highway, entering the main flow of holiday traffic. This is like the tumor cells slipping into a blood or lymph vessel. You drive for miles in the heavy holiday traffic with snowstorms, honking cars, and slippery roads. This is the circulating tumor cells surviving the dangerous bloodstream. As you approach your hometown, the highway narrows and the traffic slows down. The snow piles and you get trapped into a tight cluster, just like the tumor cells getting physically lodged in the narrow capillaries. After inching forward, you finally slip off the highway, taking the exit into the smaller local streets of your parents' town. And this matches the tumor cells exiting the bloodstream into a new tissue. Finally, you successfully arrive and settle in at home, reuniting with your family for the Christmas holiday. Except in cancer biology, colonization is the hardest part, far more difficult in our holiday road trip analogy. Now we have a general understanding of metastasis. Let's get into the scientific details of it. The first step is the local invasion and breach of the basement membrane. Basement membrane is the layer of extracellular matrix that surrounds and protects most organs. Here it would be the brown layer. The cancer cells must degrade this matrix to escape and invade the area of tissue. Through the process, the stromal cells, which is the diverse connected tissue cells around it, can assist with the invasion through remodeling the extracellular matrix. They do that by secreting factors that promote cell migration, create blood vessels, and suppress immune responses. They help cultivate the soil for the tumor cells to strive. The second step is intravasation into lymph or blood microvessels, where angiogenesis is needed. Angiogenesis is the process of forming new blood vessels from pre-existing ones. There are 3 main players to help with angiogenesis and intravasation: tumor cells, macrophages, and endothelial cells. Macrophages are white blood cells that respond to infection and injuries by engulfing and digesting pathogens, foreign materials, and cellular debris. And here, endothelial cells simply line the blood vessels. In this step, tumor cells secrete factors that break the extracellular matrix and pull the macrophages. The macrophages recruited in turn pull more tumor cells and lead to secretion of VEGF, which is a key signal needed for angiogenesis. The endothelial cells also secrete more factors that pull the tumor cells towards itself. The third step is transport through general circulation to distant sites. Tumor cells typically metastasize to the first organ en route via the bloodstream downstream from the primary tumor site. The bloodstream's flow patterns make these paths somewhat predictable. Here it shows how the heart pumps the blood through the body. The blue represents the deoxygenated blood collected from the tissues, and the red is oxygenated blood freshly acquired from the lungs to pump to the whole body. Other than the blood vessels, there are also other means of travel like along the nerves, outside of blood vessels, and through body cavities. As the cancer cells travel through the circulation, it must evade the immune system, survive the interactions with various cell types, and withstand the pressure inside the blood vessel. The fourth step is trapping of cells in tissue microvessels. The size of a tumor cells is typically 20 to 30 micrometer wide, and capillaries, on the other hand, are only about 8 micrometers. So a large fraction of cancer cells get physically trapped in the first narrow capillary they encounter. If you don't know how to imagine it, here is the tumor cells and capillary to scale. The fifth step is extravasation into the organ and formation of dormant micrometastases. Once stuck, the tumor cells must exit the vessel, survive in a completely new environment, and begin forming a pre-metastatic niche. This is essentially overlapping with the idea of seeding, which I will explain later. The last step is metastatic colonization, which is also the hardest part of metastasis. There are two parts that dictate the possibility of colonization, which are ability to survive colonization and seed a pre-metastatic niche. How cancer cells can survive colonization depends on the location of extravasation, where some tissues will be more permissive than others. For example, bone infiltration is more permissive due to fenestrated capillary structure. On the other hand, brain is the most difficult due to the blood-brain barrier and protective layer of endothelial cells, where cancer cells have a hard time to cross, and so are therapeutics. So the actual physiology of target organs does play a role in colonization, as it requires local invasion. The ability to form a metastatic colony is also needed in order to survive colonization. It is affected by factors like access to blood vessels for nutrients and oxygen. And to seed a pre-metastatic niche, primary tumors need to recruit certain types of cells to the new site before the tumor cells arrive to support and create an environment. So basically the soil for circulating cancer cells, which is the seed, to settle, survive, and grow. Despite the long journey, the odds of success for any cancer cell are very low. Tumor cells may die off in any of the above steps, but when colonization succeeds, they divide, proliferate, and eventually form a secondary tumor site. Here I'll show a short animation of the invasion metastasis cascade from the National Cancer Institute. Next, I will talk about the key contributors of metastasis, which are epithelial to mesenchymal and mesenchymal to epithelial transition. Epithelial cells are immotile, tightly bound to each other and to extracellular matrix. On the other hand, mesenchymal cells are motile and proliferative. The first part of invasion metastasis cascade relates on EMT, which allows for invasion then intravasation. Epithelial cells differentiate into mesenchymal cells, allowing invasion and metastasis. Cadherin are proteins that anchor cells, so you'll have to lose cadherin to transition. Mesenchymal to epithelial transition, on the other hand, would be the opposite. The cells transition into epithelial cells where they become more attached and less motile. It is needed for the last step of metastasis, colonization. It's important to note here that rather than a solid definition of EMT or MET, it is more of a spectrum where the cancer cells are often stuck in a partial state. So today we walk through the major steps of metastasis, but it's important to remember that what we showed here is only the surface. In reality, metastasis is shaped by incredibly complex interactions between cancer cells, immune cells, and countless signaling molecules. And cancer can behave differently. Its ability to travel, survive, and grow in a new environment can vary widely depending on the tumor type and the condition of the patient's body. Understanding these mechanisms isn't just about learning biology. It's what helps researchers develop better diagnostics and more targeted therapies. The more we learn how cancer spreads, the closer we get to stopping it. Thanks for watching, and I hope this helps you see just how challenging, fascinating, and important the study of metastasis truly is.